Let's look at the last topic of this unit, transcription and translation. So we're gonna look at how to use the DNA and turn into a protein. The DNA has the instruction. So we some textbooks say it's like a recipe to make everything you need, those proteins you need. So two main processes to produce protein. The first one is transcription, and the second one is translation. And your whole body is made of protein. So proteins has a lot of functions. This part is the review, proteins function. Protein can work as an enzyme to trigger the chemical process happens in the body. And protein can help cell contract. So this is the microfiber of the muscle, this whole structure called sarcomere. When you take AP1, you will learn this structure and how they slide, how they glide, and the whole sarcomere gets shorter. So protein help contract. And proteins help move. We talk about the cell organelles. You have the microvilli, increase the surface area, and also you have the cilia. Cilia is the one really can move. And this is a phalangula. Phalangula is one big tail that can move. So protein help move. And we also have very important proteins. They work as transporter proteins on the cell membrane. You have ion channels to make the ions go through. You have carrier, glucose carrier. And you also have the pump, sodium potassium pump, directly use ATP as energy source to move the molecules. And also in the blood, we have the lipoprotein. You have the low density one, it's called LDL, low density lipoprotein. You have the HDL, high density lipoprotein. So they are used to transport. And this is the protein in your blood to carry oxygen. That's the hemoglobin. And proteins can work against pathogen. So this is antibody. Antibody is a protein produced by one of your lymphocyte. That's one of the white blood cell belong to the lymphatic system, and they can target the pathogen. And also you have receptor proteins help cell to communicate. Like when your body release a hormone, say release insulin, and insulin's job is to bind with the insulin receptor. And once it binds with the insulin receptor, it's the receptor protein. It will trigger more glucose carrier put on the surface. So you have more glucose carrier that can carry glucose into the cell. And so outside the cell, in the blood, glucose level decrease. That's how insulin decrease the blood sugar level after you eat a lot of carbohydrate. So hormones need to bind with the receptor, and receptor is one of the protein's function. And protein need to maintain its 3D structure to perform its function. That's why when we talk about the protein synthesis, it's not just one organelle. You need to go to rough ER, from the rough ER, and you create the amino acid sequence, and then transport it to Golgi apparatus to fold it to the correct 3D structure. So every protein needs to maintain its 3D structure and to perform its function. And when the 3D structure could not be maintained, we call it denature, right? So what decides the 3D structure of the protein is a 1D structure. All the proteins are made of amino acid, and there are totally 20 different amino acids. And some of them are hydrophilic, they like water. Others are hydrophobic. So when you put an amino acid sequence made of those different kind of amino acids, when they fold to a correct 3D structure, the hydrophilic one, they don't mind facing the water. But the hydrophobic one, they will mind. They will be folded inside, so they won't touch the water. So they decide the 3D structure. The 1D structure of amino acid sequence decide the 3D structure of the protein. So if the amino acid sequence is wrong, then the protein structure will be wrong. And then today's topic, okay, what decide the 1D structure of amino acid? That's the DNA structure. So if the DNA is wrong, eventually the amino acid will be wrong. So that's the key concept before we start. So okay, why transcription, translation, they are important? Because if the process goes wrong, and eventually the amino acid sequence will be wrong, and then the protein you produce will be wrong. So there are totally 20 different kind of amino acids. And those amino acid is called amino acid because it has an amino group. This is amino group. And this is carbolic acid group. So together it's called amino acid. Okay, so DNA carry the instructions to produce O protein. And then from the DNA, before they produce protein, let's review what they need to produce. RNA, right? Or more specifically, mRNA, from the DNA to mRNA. And there are totally 3 kinds of RNA. So mRNA is one. It carries the information from the DNA. And there are other 2 called tRNA and rRNA. So totally there are 3. There's 3 different kind of RNA. So mRNA is the one carry genetic information from DNA. It's like, okay, you write down the note from the DNA. And it's made of A, U, G, C, those 4 nucleic acids. So they are written recipe from the DNA. And tRNA is called transporter. So tRNA's job is to carry amino acid. And you read mRNA. So tRNA need to be able to read, know which amino acid is supposed to carry. Each tRNA only carry 1. So those mRNA, they have a sequence, the sequence called codon. Today we'll talk about it. Every 3 is called a codon. So the tRNA actually have the match one. It's called anti-codon. So it will match the mRNA. So there will be no mistake. You know, okay, there's 3 codon, match those 3 anti-codon. So together they will bring this amino acid number 5 here. So that's tRNA's job. And the last RNA is called rRNA. R means ribosomal. So on the ribosome, there's the place for the translation to happen. And a big part of ribosome is actually made of rRNA. So that's the ribosomal RNA. Totally, there's 3 kinds of RNA. And we talked about these slides before. This string is the mRNA. And once you have mRNA from the nucleus, go into rough ER. In the rough ER, it's called rough ER because they have ribosome. So those ribosome will attach. They have 2 units, big unit, small unit, like 2 magnet. They will glue. They will attach to the mRNA. And now they start the translation. So translation happens in the rough ER with the help of ribosomes. So ribosomes, and a big part of ribosome is rRNA. So they can help the translation process. So these 3 kind of RNA, messenger RNA, ribosomal RNA, and transfer or transporter RNA. So there are 3 kinds of RNA. So far it's a review. And RNA, the mRNA is a single strand. So it's just carry the information from DNA. So it's not as stable as DNA. So DNA is very stable, not RNA. Okay, and also let's compare the DNA with RNA. What's the similarity? What's the difference? Also, that's the review. So similarity, they are made of nucleic acid. And there are 5 different kind of nucleic acid. For the DNA, use A, T, G, C. RNA use A, U, G, and C. And so they are like 75% identical, only 1 difference, right? And DNA, A pair with T, G pair with C. And for the RNA, they use AUGC, single strand. And their sugar is ribose. Ribose, that's the RNA. Okay, so that's the DNA, that's the RNA. Okay, here it show you the DNA, the sugar use deoxyribose, or you can say 2 deoxyribose. And RNA use the ribose. Okay, that's the difference between DNA and RNA. Okay, so that's the DNA and there's the RNA. And that's the RNA nucleotide. You have a base, you have a ribose sugar, and you have 1 phosphate. And that's the sugar for ribose that's used for RNA. And this is 2 deoxyribose, and that's used for DNA. Okay, so far, these are the review. And let's take a break. We'll start to talk about the real transcription and translation.